We've got Peter Boutsalis joining us. Peter is going to speak to us about understanding glyphosate resistance. He's the right person for this job because not only does he own plant science consulting where he does lots of herbicide resistance testing, but he's been a researcher at the University of Adelaide and working with a team of people there who understand glyphosate resistance. And also Peter has been involved in the random surveys around South Australia and Victoria, sampling weeds and periodically every year to check the levels of herbicide resistance around the country. So he's seen thousands and thousands of pots, some of which are glyphosate resistant and some are not. So we're going to hear more about that. So good morning, Peter, and thanks for joining us.
G'day, yeah, I'm on. That's good. All right,
all good. Ellen will hand over controls to you, Pete, and once she does that, we'll be able to hear from you, and you can launch into your presentation, and I'll interject periodically, and we'll have a few questions as we go.
Okay, I'll just try and move this go to webinar control panel to the side a little bit. It's perfect. Okay, so can everyone hopefully can see that presentation that's going through now?
Yeah, it's all good, mate. Yeah.
We're good? Okay, here we go. Good morning, everybody. And so just to start up, a little bit on what's been happening with glyphosate resistance in Australia, both in the winter and the summer sector. Glyphosate resistance was first identified in 1996 in annual ryegrass, and since that time she's gone up exponentially with thousands of populations. You know, they're the ones that we've confirmed there, but there's thousands more, and at some stage it's not exciting anymore to count glyphosate resistant populations. There are other species in the winter weeds area. The wild radish is slowly coming onto the scene, and a couple of brome species as well. So there are some other species also that are coming in. To add to that one as well, I know it's on the other one there, south thistle, which occurs both in the winter weeds area and also in the summer weeds area. So in the summer weeds we've got barnyard grass, which has been a leader. Fleabane is also increasing. You know, fleabane's tolerant to glyphosate at lower rates, but at high rates glyphosate kills fleabane. But these populations are actually glyphosate resistant. Where they've done the mechanism work, they found that it actually has a resistant mechanism. Lever seed grass is also, I think there's a lot of populations there, we just haven't had many to confirm it, but it's there. Windmill grass as well is one of those other ones like fleabane that's quite tolerant to glyphosate, but spraying high rates you can kill windmill grass, but yeah, these guys just healthy as. And south thistle, of course, that's the latest one coming on, and that's a bit of a worry because south thistle's present throughout the entire country, both you know, in the northern part, in the summer growing and the winter growing area, where we've tested south thistle for resistance to group B herbicides. 90% of it is resistant to group B herbicides. We're just starting to detect glyphosate resistance in south thistle and also detecting 2,4-D resistance in south thistle. So group I resistance. So that one is a real worry because there's lots of, very important modes of action. Okay, that's a bit of a summary. So that's some table what we've confirmed. We said that earlier, and just to give you some numbers there.
Pete, there was a question already about how much resistance in brome grass has been found, and you've got, I think, on the previous slide, 5 populations. Would I be right in guessing it's quite a bit more than 5 now, or is it still pretty good number?
No, we've only confirmed those 5, so we don't know of any others. Yeah, there could be 1 or 2 more, but that's about it.
Righto.
So yeah, they've come mainly from sort of fence line areas where there's been repeated use of glyphosate without the addition of, you know, having a crop to compete and slow down the process. But yeah, that's a bit of a worry, that one. So most of the glyphosate resistance is in ryegrass is occurring in winter grain, so in our cereal crops, and these are not fence line, these are within the crop, so because of repeated use of the herbicide. The other main areas there are from fence lines or crop margins where you've got repeated use of glyphosate for many years, and there hasn't been a lot of other herbicides used, and also there's no competition with crops. So, where you may have a situation in the paddock where you've got a weak survivor, and we'll talk about weak survivors soon, in a competitive crop situation, you can now compete that weed, even kill it. But in a fence line, once everything is dead and you've got a very weak survivor, everything else around it is dead, that guy can produce thousands of seeds at the end of the season. The other one as well is roadsides, and that's where Uni of Adelaide did a random survey along roadsides throughout all our in Adelaide, Barossa Valley, Adelaide Hills, Clare Valley, and found that almost all of the ryegrass that they identified was glyphosate resistant. So that's a bit of a worry as well, because we've got glyphosate resistant ryegrass occurring along roadsides. The common thing there has been about 15 years of continuous, more or less continuous use with very little additional herbicides. Okay, now what about randomly? What's happening randomly? As Pete mentioned earlier, we've been doing these random weed surveys throughout South Australia and Victoria every year, visiting the same region on a 5-year basis, and we're really looked at thousands of paddocks there. The new GRDC round of funding, which occurred recently, really has identified that glyphosate needs to be tested as well, and so we started looking at glyphosate, and more or less in all areas we're just starting to see the odd population coming through. Now in these surveys, if you can detect any form of resistance, it's getting a bit concerning, because we are picking these paddocks totally at random, and we really only know 10 seconds before we get to the paddock which one we're actually going to visit. Concerning the southeast of South Australia, where 16% of the samples were glyphosate resistant, I didn't believe that initially, so I repeated the test with several rates and found exactly the same result. So that's very significant. Last year we surveyed the Victorian Mallee and the Wimmera, and there we found in the Wimmera 9% and Mallee 3% glyphosate resistance. So that's quite concerning. So we are picking it up now in the random weed surveys. So that is, it means that it's happening out there.
Can you tell us a little bit about the farming systems in those areas that is contributing to that resistance?
The farming systems, well...
So in the southeast, is it that pasture seed industry that's quite famous for developing resistance, or is it all over?
Oh, it's all over, definitely all over, yeah. Right. Down the lower part of the southeast, we've got that sort of white clover, lucerne type areas, but we also had a lot of paddocks, cereal paddocks in the upper bit of that survey also. So yeah, really occurring everywhere, Pete. Even in all of those areas that we target are intensively cropped areas. We're not going into areas which are just pasture only. Okay, I'll move on. That's just a photo to show you the latest survey we did in the Wimmera, and that's what each pot there represents a paddock that we visited, and the green pots there are where we've identified glyphosate resistant ryegrass. Now what we call resistant is if we have 20% survival or greater in that pot test. So if there's any samples in there where the percent survival in that pot was less than 20%, we don't include that as resistant. So all those numbers I showed in the previous slide was the incidence of paddocks that had glyphosate resistant ryegrass greater than 20% identified in our pot trials. Just a bit of a photo there. So let's see some facts about glyphosate resistant weeds. Now all growth stages are resistant. That's important. So whether it's a seedling, it's a large tillering plant, or the pollen, they all contain, because the resistance is transferable. Younger seedlings seem to be more sensitive than older plants, and so that's important to know. Resistance, yep, it throughout all the stages of the plant, and because ryegrass is an obligate outcrosser, it must receive pollen from another plant to fertilized seed. So if you've got two resistant plants next to each other, well, what's going to happen is the progeny will be more resistant, and so increase the level of resistance. So you need higher and higher rates to kill those plants if that works. So that's a special attribute that ryegrass has, and also wild radish also has that cross-pollinating attribute. We've detected where we've identified fence line resistance. We've gone out to 50 metres and surveyed and found that even right out there, as we went out in the paddock, the resistance decreased, which was indicating there was probably coming from the fence line, but we could detect it to 50 metres out, so it does move in the paddock as well. So don't think that if you get resistance, it's just confined to the fence line. We've heard a little bit about the fitness of ryegrass, glyphosate-resistant ryegrass, and where we've looked, some of these populations seem to be less fit, so they can't compete with susceptible ryegrass as efficiently in the absence of glyphosate. If you stop using glyphosate, then the resistance it has does cause the plant to not be able to compete as well. And basically what that means is that these guys produce less seed. So if in a situation where glyphosate is not used, then over a period of 4 or 5 years, you can actually crash that population right down to almost no resistant seed. That's happening, we reckon, in quite a few cases. But as glyphosate resistance evolves and ryegrass evolves, then we believe that we'll find populations which have got the same fitness as non-resistant ryegrass. So we haven't confirmed it, but that's where we think the direction that we're going into. Okay, so this is just to indicate again that cross-pollination. So this graph here shows 3 different biotypes of ryegrass. We've got susceptible, we have a TS, which is a ryegrass resistance, glyphosate target site resistance, and we've got a translocation mechanism, and we'll talk about that mechanism as well. So just like with group Bs and group As, we can get target site resistance to glyphosate in ryegrass as well. Now where we've found it, it doesn't occur very often, and where we found it, you might think the resistance would be really high, but it actually isn't. So the target site resistance to glyphosate actually gives pretty weak resistance, unlike most group Bs and group A target site resistance. Now where we crossed the target site resistant population with the translocation resistant population, we got population that's in the dotted line that you can see is much more resistant to glyphosate. So 2 mechanisms in the same plant, and that's what happens. We've also found that in a couple of the populations as well, especially from some of the vineyards. And so yeah, it does occur in nature as well. We not just in the lab, but we have identified it in populations out there.
This is a pretty critical bit of information, isn't it, Pete? I've heard agronomists say to me that they feel that the ryegrass has just been getting that bit more resistant year after year, and they've been putting the rates up year after year to sort of keep in front of it. And I think this slide here would back up those observations, wouldn't it? That the ryegrass got a bit of resistance, higher rates killed it, but then it accumulates more genes and it gets more and more resistant.
That's right. So we've got two different mechanisms here, but also ryegrass is a diploid, so it's got 2 copies of every gene, and so you can have a situation where you've got one of the genes is target site resistant and one isn't. If you've got both of the genes, that means that the resistance will be higher, and that goes the same with the translocation mechanism. So you can have a few combinations of permutations which gives you different levels of resistance as well. Another thing I've been thinking about is this translocation mechanism. It's not as clear cut as, say, the target site, where it's an exact amino acid substitution that we can identify. The translocation mechanism, we believe there's probably different forms of it, and depending on if you've got plants with slightly different forms, they can probably accumulate, and so you have a higher level of that translocation mechanism, because we've got plants that will survive very high rates of glyphosate and they have the translocation mechanism. And we've got plants that are very weak, have weak resistance, and they have the translocation mechanism. So you think they've got the same mechanism, but their resistance levels are so much higher. So there must be different forms of this mechanism, or possibly another unknown mechanism as well. So there's lots of, yeah, a lot we don't know, but it's becoming quite important. So I'll just move on because I've got a bit more information on the next slide about these mechanisms. So where we, reduce glyphosate efficacy, one reason is resistance. Depends on the mechanism. Now this translocation mechanism, what happens in that one is that the in-resistant ryegrass, it gets moved to the leaf tips and doesn't translocate, move throughout the rest of the plant. The plant requires a lot of energy to do that, to keep the glyphosate right up there, and so that could be one reason why there's reduced fitness, because it needs a lot of energy to move that glyphosate right up in the leaf tips and keep it there too.
So are we seeing a symptom of that, Pete? You see burnt leaf tips with that?
Yeah, absolutely, burnt leaf. Yep, the older leaves are burnt, and but as you increase the rates, then those active pumps just can't get rid of all that glyphosate, and some of it starts to leak and starts to cause some damage because you get sublethal rates going to the root zone and the growing points, and so you start to really damage the plant. And that's why higher rates often will improve control. Yep, the target site one, we've talked about that one, but it's a single position. It's a position on that gene, the EPSP synthase gene. That's the target site for glyphosate, and it's a position 106. And normally there's a proline, and there's a substitution to serine or threonine. When you get that substitution, it gives you that resistance that we talked about. You can get both in the same plant in ryegrass as well, and we've spoken about that. There is another mechanism that we found in our Australian brome, but also occurs in a couple of the Amaranthus species in the US, and that is the increased expression of that gene, that enzyme, the glyphosate, where glyphosate binds. So where you have, in these particular resistant populations, the enzyme is susceptible, but you've got 1,000 more fold copies, and so you add the glyphosate will inhibit some of that enzyme, but there's enough enzyme left for the plant to keep on growing. And so that's where we've got that third mechanism. Haven't found it in ryegrass so far, to the best of my knowledge, but it could be out there as well. It's funny that
you mentioned earlier that the 5 bromegrass populations, have you confirmed that they're all that gene amplification or the increased expression, or you're not sure yet?
Where we've looked, it is that mechanism, yes, where we've looked. I don't know if we've looked at all 5, but I think about at least 3 that we've looked have got that mechanism. So that's very interesting that different species has a completely different mechanism than ryegrass.
Okay, everyone listening in out there, make sure you put some questions in. We'll stop for questions in a moment, but yeah, anything that comes up, just type in a question and we'll pause in a minute and do a few questions at once.
Okay, so that's where we've reduced the glyphosate efficacy because of resistance, but that's not always the reason. So here we have other factors. Now, treating older leaves, older plants, larger plants, e.g. the sort of situation you see on fence lines in spring, which has been a very common time to clean up the fence lines. Those plants are also a lot more resilient, and also you get poor coverage because you've got such a massive ryegrass that you just physically can't contact every bit of leaf, and the rate of translocation that occurs, you don't get enough into that plant, and that can be a sort of a sublethal rate. Something similar to that was the first case of paraquat resistant in ryegrass they identified in South Africa, was in orchards where they were spraying really high rates of paraquat on very large plants. And so what was happening, this is in ryegrass, so what was happening is that they were getting a sublethal rate because they weren't getting, proper coverage, and they slowly started to select for paraquat resistance. So this is probably one of the reasons why we've got that resistance. Okay, nozzles. Are your nozzles putting out the correct amount of spray, water rates, the right calibration, the correct boom height? They're all factors that will determine if you're getting the right rate, the rate that you think you're putting on or not. Additionally to that, stressed plants. Plants are stressed and they don't get the same uptake. Stress can be due to temperature extremes, drought, nutrition, poor nutrition, pest damage. They all affect the translocation, the movement of nutrients through the plant, and also the translocation of glyphosate through the plant as well. So that's reduced, and so you can get plants surviving due to that. We've also identified that at higher temperatures, glyphosate doesn't work as well as in cooler temperatures. So if you spray plants at 30 degrees, you get less effect if you're using the same rate. If you use it at 20 degrees, you'll get much better control than you will at 30 degrees. And it's a pure temperature effect, and we've identified that, and also ARI have identified that in barnyard grass, that at lower temperatures you get a lot better, glyphosate works a lot better, and we're not exactly sure why that is. Also, as most of you would know, that dust combines to glyphosate, and so you get less effect, you get less herbicide into the plant. So if you've got a dusty situation, some of that glyphosate is going to be bound by the glyphosate, by the dust, and so you get less effect. So it's like a sublethal effect. So they're the non-herbicide reasons.
Yeah, we've got a few questions come in, and they're a little bit, some of them are a bit relevant to this, so we'll pause and do a few questions. And one of them, Glenn Reithmuller's put up saying, I thought Dave Minke did some work and showed 8 litres per hectare of glyphosate didn't kill ryegrass but were not resistant, and it was due to the waxy leaf from early water stress. Dave, if you happen to be listening, feel free to put up a comment in your questions box now. The other thing I remember from Dave's work was he did a trial, I think, at Albany on nitrogen deficient ryegrass, and he couldn't kill nitrogen deficient ryegrass with higher rates of glyphosate either. So yeah, any comments there, Pete?
Yeah, I don't have that kind of data, but that ties in with what I've been saying, that stressed plants, you just don't get that herbicide moving to the active sites. I think it's
and so
I
know John Moore's out there.
That information.
John Moore is listening in, and he's been involved in the Herbie Rate program with Dave Minke as well. So John, you might like to chuck a comment in the question box as well about that. And I seem to remember from that work that when you have that moisture stress and heat, the plants do have the waxy leaves, and then when the weather comes good and we get some rain and cooler conditions, we really do need to wait for the new leaves to grow so that they don't have the thick waxy cuticles, so we can get uptake.
Yep.
Okay, so here's a bit of a loaded question, and I'll name it. It's from John Moore, and I know that John has been quite vocal about this. He says, should we eradicate brome populations, glyphosate-resistant brome populations, when we find them? So John's been sort of taking a biosecurity approach to this, saying, well, when we've got this new thing, it's like a new species, should we treat it like an exotic weed and try and eradicate it? So let's do a poll now. Anyone out there who thinks yes or no, should we try and eradicate brome grass as we find it that's resistant to glyphosate, keeping in mind that it will cost money to do this and we will need funding to do it? Put in a yes or a no in your questions box now, and we'll have to do a quick poll. But yeah, what are your thoughts about that, Pete? Should we treat it like a noxious weed?
Yeah, look, it's very difficult to do that, but it may not be impossible. It just means that brome has got a dormancy, and so you have to isolate the area for several years, and whether that, depending on what your soil type's like, what your tillage program's been like, also just the inherent specifics of the particular biotype, it could take even up to 4, 5, 6 years to be on top of that. So that zone has to be out for that period in time. And if someone's prepared to do that, then you could, in theory, eradicate it from a particular section of a paddock, especially if it only occurs in a small area. One of the first brome that we found, the glyphosate-resistant brome, was in the middle of a paddock, but that middle of the paddock was an old fence line, and so the brome ran, the resistant brome ran in the same direction as the old fence line. When we collected, because we identified it was identified as plants, and so we took plants from that sort of old fence line area, and also plants 10 metres from the area, and those plants were entirely susceptible. So in this particular case, the brome was really defined to that sort of old fence line strip in the paddock, and it hadn't moved out too much. So if you wanted to really, bomb that zone, you could in theory destroy that brome from that particular paddock. So it takes a lot of effort, but it's not impossible. Now remember that this resistance is occurring, out there at random in different areas, and so there may be growers out there that have got the issue but haven't realised that they've got resistant brome. So it could be so you can probably, if you talk about eradication from a particular patch in a paddock, I think that can be done. But to say we'll do it out of an entire state could be a bit of an issue. But you have to start somewhere.
Okay, so in the poll we've got about 6 yeses, I think, and about 5 no's. And a couple of the no's are quite vocal about how they think it's a bad idea because it's sort of it's bringing about like a legislation type thing where growers would need to be quarantined and so on. And so yeah, it's a sort of a 50-50 answer, that one.
Sounds like the election on Saturday we're going to have.
Yeah, let's not talk about that. Okay. And so one of the comments there was the moldboard plough can fix that in 1 year. But, it doesn't quite fix it in 1 year. But, yeah, I mean, people could themselves take aggressive measures to eradicate a patch on their farm, and many farmers do. But, yeah, I guess the question is, do we want it to be like a compulsory sort of thing that people have to do and involving quarantine, and that's probably why that one gets a range of yeses and nos. All right, another question.
You'd have to, just before we go, you'd have to actually in those areas continue spraying glyphosate to determine if you've got any survivors, and those survivors then would need to be handled with, say, a high rate of paraquat or something else.
Yeah, I think you'd have to keep eradicating it until there was zero seed bank, really, wouldn't you?
Yep.
Now, here's another one that comes up all the time. What is the ideal water rate to spray glyphosate on ryegrass? Now, there's some old work showing that low water rates were good because you get this high concentration in the droplet and so on. But I think, personally, that that's a small effect, and there's a big confirmation bias out there. People want to hear that 30 litres of water is good, but what are your thoughts, Pete?
Yeah, I was looking at, the other day, I was looking through the old Roundup Palmax label, and it said in the label that at about 70 litres, you've got the correct amount of inbuilt adjuvant. Whereas if you go higher rates, and I think it was 70 off the top of my head, if you go higher than 70, you're going to need to put some more adjuvant in there because you start getting a bit of a dilution effect. So yeah, I reckon about you sort of if you go higher, you may require to add a bit more adjuvant to get that proper wetting effect.
Yeah, but there's also this thing about the concentration gradient, and that a high concentration of glyphosate in the droplet can be a good thing. Are you aware of that work or not?
Not specifically, no, I'm not. But yeah, you need to have, as long as you can ensure coverage on all you target with a lower concentration, then that would do the job. But I don't know of the specifics of that research, no.
Yeah, and I know John Moore once again is out there, and you've got some opinions on this, Maury, so you might like to put them up now, and I'll get to them in a minute. But I think obviously what we have now is we have glyphosate rarely goes out on its own. It's often with another herbicide, and we often need plenty of water for the other herbicide anyway. So yeah, it's not a huge issue, I believe. Now, a question that I think you've already answered, Pete, but we'll go back to it. Does spray topping for seed set control of glyphosate-resistant rye or brome grass work, considering that it's sequestration at the tip, e.g., does it go to the seed head? Yeah, thoughts on that?
Oh, okay. That's a good question. You know, using it like that. We've found that it's in the genetics, it's in the pollen, and so would plants with translocation mechanism be resistant or not? What we've sort of known is that if it's resistant at the whole plant level, it's resistant at the pollen level. But that's something I've got to think about a bit more, and yeah, I'm not exactly sure on that one. The target site one would be, definitely, where we had looked at target site resistance to other herbicides that were in to group A's in pollen. This is 30 years ago, but we found out the pollen was resistant, where we had germinated the pollen and found that the pollen was growing in the presence of herbicide from resistant plants, but susceptible pollen wasn't germinating. That was with group A's. I'd say it's the same for target site resistance to glyphosate, but the translocation mechanism, I couldn't say yes or no.
Okay.
That's right. Probably an area that we need to look into.
Yeah, that sounds good. All right, we'll jot that one down.
Especially it being the main mechanism out there.
Yeah, that's right. So reduced translocation is the #1 mechanism in ryegrass, isn't it?
Yep.
Okay, so we need to keep moving. We'll bash through a couple more questions and then we'll get going. Does dust from tractors in dry fallow neutralise glyphosate?
So if it's on the leaves, you get some of that glyphosate irreversibly binding to those particles, and so you get a sublethal effect if it's a lot of dust.
Yep. And another question, what about the use of adjuvants to improve the efficacy of glyphosate, e.g. for waxy leaves?
Yep, no, definitely the case. Things like organosilicon adjuvants like Pulse is one. They improve control. So yes, you can get improved control with some of those adjuvants. And Pulse is one where it's used in controlling perennial woody type plants, or plants or weeds with very difficult cuticles to enter. Something like Pulse will help that glyphosate get through and then do the damage.
Yeah. Okay, we've got some other good comments in here about spray coverage and water rates. A few different people have come in. John Moore has said that water rate is really a function of the equipment rather than the chemical. We've got another comment that 50 to 100 litres of water per hectare, higher concentration is definitely good, but in general rates have gone up a lot since that work. And another comment, we need to get 6 to 8% coverage for glyphosate to work effectively. At 30 litres you will have a high proportion of fine droplets and you are introducing another drift problem. Yeah, so that's all good comments there. And there's lots more questions here about adjuvants, Pete. So lots of questions about could adjuvants, weta TX, ammonium sulfate, the best adjuvant package, could they help overcome glyphosate resistance in any way?
Yeah, well, where they would help is where you're at the beginning of a situation where you've got weak resistance in the paddock. So as we know, you increase the rate, you get better control. Well, if you can, by any way, increase the efficacy of glyphosate, then that would help. Where it wouldn't help is if you're already in a situation where you've got quite significant levels of resistance, then yeah, adjuvant won't really do very much. But if you're at the beginning, and I'd say most people, if they have any kind of resistance, would be at the beginning, then yes, it would help in those situations.
Yeah, and from my experience, I think with other herbicides as well, when the herbicide is really working well, we don't have much resistance in the populations, We could get it wrong and still kill the weeds. We can have the wrong adjuvant, the wrong water rate and everything, but the weeds are so susceptible you still kill them. But as we get resistance to these things, you have to get everything right. You have to get the water rate right, the conditions, the adjuvant, you know, the plant can't be stressed. It has to be all going well to overcome a resistant weed. And so, yeah, it all matters, but it doesn't fix the problem, I think. Yeah. Okay, Pete, we'll keep on going there. We've interrupted you a bit. We'll let you plough on for a while, get through some more slides and do some more questions.
No, it flows through nicely. This is so far this year through the quick test, I've received, well, I've reported 20 ryegrass samples that have come from, you know, in paddock knockdown situations, and of those that were suspected of being glyphosate resistant, 14 were not resistant and 6 were resistant, so sort of, you know, 2/3, 1/3 almost. So it's not always glyphosate resistant. That photo there is a paddock at Roseworthy that I visited about 4 years ago, where the farmer there had sprayed glyphosate, and there were some true survivors. And so I took the photo to represent a situation where you've got, you know, real suspicious resistant ryegrass. I took those surviving plants, those healthy plants you see there, I took those back to the lab and did a quick test, and I killed them all with a litre of glyphosate. So up to this day, I'm scratching my head as to why they were not resistant. So it's not always, even if you've got a situation that's as clear as this, you just can't be guaranteed that it is glyphosate resistance. And I think this photo just demonstrates that exact situation. Okay, this is some data I've got from the farmer testing. It's mostly seeds or plants as well. So what I've done there, I've got there, they are populations where we've confirmed resistance from all over the country. I've ranked them accordingly to the resistance to 1,000 litres of glyphosate, sorry, 1,000 mils of glyphosate, so 1 litre of glyphosate. And just want to demonstrate there that where you've got low level resistance at a litre, it doesn't always mean that higher rates will control it. So if we look at that third line from the top where I've got that blue, this blue, if you can see that, here is a population where we had 5% survival at 1,000 mils, but as we increased the rate, we got no improvement in control. Down here, this population from Western Australia, we had 80% survival a litre, and increasing the rate more or less did nothing there. If we, just to change the scenario, when we went down to, you know, this particular one from Bannockburn in Victoria, all the plants were highly resistant at a litre per hectare, but as we increased to 1.5, we killed everything. So there's no real correlation to, if you've got a low percent surviving, then that you're going to control them with a higher rate. Yes or no, we just don't know. And that's what I've pulled in a lot of populations there to try and just give a bit of an indication, you know, what's happening. So we've got, you know, some populations up here where we've got very low levels surviving a litre, but increasing the rate, you know, you don't always improve the kill. And other situations where you have a lot of survivors, but increasing the rate, you get total kill, complete death. So it's up and down all the time, and that's due to all the, you know, different mechanisms coming in together. Okay, so we demonstrated that one. So how do we improve glyphosate efficacy? Higher rates, as I've mentioned several times, they can aid in buffering the effects of some of those non-herbicide effects, such as older leaves, older plants, stressed plants, spraying in high temperatures, poor coverage, dust. If you increase the rate, you can actually overcome some of these factors. So a bit of an insurance policy, just in case there's a factor there that's not really, it's going to reduce the effect of the herbicide. Let's put in a bit more glyphosate. It's cheap. Put in a bit more and get better kill. testing to know if you've got resistance is important. So whether you're in a situation where you've got plants in the paddock or at the end of the season, seeds, just to know, do you have resistance and will a higher rate control those resistant individuals or not. Avoiding any survivors to cross-pollinate, because as we've said, they will accumulate that strength of the resistance level, and so it'll be harder to kill plants with the rates that you've been using the following season. And even if glyphosate is working well for you, then really do something else. Don't rely on it. Paraquat products along the fence line, use some other products as well. Don't only rely on glyphosate. Mix it up and try and get those plants being controlled. So I've got a few tests, actual summary tests of farmers that I've received I've done some glyphosate work for, and this is one from Elmore that I reported recently, just last week, I think the week before. And in this situation here, excuse me, the farmer suggested that he thought that factors other than herbicide resistance, he didn't think he had resistance, but he reckoned something was wrong, and the test confirmed that. The test confirmed that with glyphosate 540 at 1.5 litres, that there was no survivors. So, what he thought actually happened, that his plants weren't resistant. Another farmer from Lake Bolac in Victoria, this was in a knockdown situation, and he tested 3 rates, and what he found that at 1 and 1.5 litres there was no improvement in control. No improvement in control, whereas increasing it to 2 litres, he got only 10% surviving, and those plants had quite weak resistance, so they were very damaged, but they were recovering, and we'll look at some photos of those in a minute. So better control, but those weak plants, if they're allowed to set seed, then that's just going to worsen the situation for next time. A farmer from Bordertown, he suggested that he thought it was shading from young seedlings. He had a lot of stubble, and he said that he reckons that it wasn't resistance, and the test showed that with 2 litres he got no survival. So he was right that it was shading was probably the reason. Now if we'd used 1 litre in this test, we don't know, he may have had low level resistance that was overcome by 2 litres but not 1 litre, we're not sure. But at least what he thought, I believe, is the correct finding there. Another one from Cummins. He, in this situation here, glyphosate survived in the paddock after a knockdown. 2 litres did the job on that. So farmer can be confident that at least at higher rates of glyphosate, there's no resistance to that as well. A couple from WA. This one here, again, this was from a knockdown paddock, and the farmer tested quite a few herbicides, but to glyphosate he used 3 rates and found that at 1 and 1.5 litres there was survival, and there was actually no improvement in control going from 1 to 1.5 litres. With the RR rating there indicates that those plants were quite healthy. They were stunted, but they were recovering. Whereas 3 litres, yeah, there was no survivors left there. I think it's the last one. This is from Esperance, and the grower suspected poor application, and he was correct, because a litre of glyphosate, there was no survival. So again, just to help verify some of the thoughts that the farmers have got, the growers have got during the season, just to know that you suspect something. It's very easy to verify whether that's the case or not by doing a simple test. Okay, so just to summarise some of the results from last year. So I've had 4 rates, 1, 1.5, 2 and 3 litres of glyphosate, and growers have got an option to choose 1 or more of those, or even if they want another rate, that you can do that as well. So at 1 litre, 200 of those ryegrass have been treated with a litre, and 29%, so almost a third of those survived. At 1.5 litres, we had 25% surviving, so a little bit less. At 2 litres, we tested 100 ryegrass samples, and 15% survived. And 3 litres to 3 litres of glyphosate, again, quite a few samples were tested, 125, and 10% survive that. So it just goes to show that at increasing rates, you get fewer populations actually surviving. So this is a different way of presenting that theory.
Yeah, that's a critical piece of info there, isn't it? And yeah, you're going to go to that slide again, which really does sort of show what the agronomists are suspecting in the field. That the first time they see glyphosate resistance, it might be a low-level resistance, and that can be overcome by higher rates in quite a few instances.
Yep. So just to bring in that slide again, that just shows that, you know, at higher rate, as you increase the rate, you can kill the target site one, the translocation one. We'll almost killed it at, you know, 2,000 grams of glyphosate, so almost sort of 4-ish litres. But yeah, at lower rates you get a lot more surviving. So there's a lot of differences within a single population of level with respect to level of resistance. Okay, I've got some photos here just to demonstrate some of that. Top photo, we've got resistance at a litre. All the plants there were surviving at a litre per hectare, whereas at 1.5 litres we got complete control. Down the bottom, this one's, you know, killed susceptible situation where even a litre should do the job. So it's really important to use the highest rate that you can sort of afford, because you're going to get a lot better kill of any weak resistant ones. And imagine the ones that have survived the litre up the top photo, all of those have survived. They're going to set seed, and that could be a disaster next year. This is a photo I took just yesterday, and that is the back row is one population, back left is untreated, back middle is a litre of glyphosate, and the right is 3 litres of glyphosate. And really there's not a lot of improvement by increasing it to 3 litres. You've got a bit better kill, but there's still quite a few plants there that are going to set a lot of seed if they're let loose and, you know, don't have anything else done to them. The front pot there just shows what a litre of glyphosate should really do, and that's the bit of the doornail. Weak and strong glyphosate resistance from the same paddock. So this is one pot from the same paddock. You can see the plant on the left is heavily damaged. You've got real curling and it's all sorts. It's recovering, it's producing shoots, and in a couple of weeks' time that will look very, very healthy. The plant on the right is healthy as. They're from the same paddock, so you've got, you know, 2 different responses occurring in the same paddock. And even right on the back there, I just noticed that there's one there, that's another plant back there, and that's really low level, you know, that could be low level resistance there, even lower. So you could have 3 different types of resistance in the same paddock, just in that little pot there.
So essentially, as we keep going, we select for those more resistant ones, don't we?
That's right. That's right. And this is just a close-up. So this guy can, you know, cross-pollinate with another weak survivor if they get to that pollination stage, and just boom up that resistance level to much higher rates. So they survive high rates of herbicide. But you can see the glyphosate's really doing damage. So it's tried to, you know, if that's a translocation mechanism, it's moved some of that glyphosate out, but it just couldn't do it all, and yeah, you've got that situation. How do we control weeds on the fence line? Well, very common practice is to do that right at the end of the season when there's not much to do. You know, your footy team like mine's not going too well, and sort of you've given up. So get out on the tractor and spray some fence lines. And this particular fence line in Clare, north part of the mid-north there, 2 litres of glyphosate has really done nothing. You know, you're trying to walk into a wall-to-wall ryegrass and expecting a lot for a herbicide, and so this is highly resistant, this particular paddock fence line. We've got some yellow there, or bromacil, so Uragan from Adama, and paraquat mixes doing a really good job, and these guys were dead as a doornail. This is only 2 weeks after spraying. So these guys were dead as a doornail, and so a lot of different effects. But if you think about it, you're asking a lot from a herbicide to try and really do a job there. If you come in we've done trials where we've come in very early, so before sowing, just after sowing, and put in some residuals along the fence line. You can really minimise what's going to germinate through, and then come this time of year, if there's plants coming through, it's a lot easier to control 10 or 20 ryegrass plants per square metre than it is to control 2,000 plants per square metre, which is what was happening at this fence line. Sowing right along the fence line as close as you can, you then put in some crop, and that helps to compete with any potential survivors as well. Having a boom right on the edge of I don't know the exact terminology, but having a separate line, so when you do that outer lap, you can really put out a high rate of a residual herbicide to get that right along the crop margin, and that's very effective as well. So anything that can reduce the number of plants that are going to be exposed to glyphosate will help reduce the risk of resistance occurring too quickly.
There's a few people having a go at you about the drift into the neighbouring crop there, Pete.
Yeah, that's funny. That is actually, believe it or not, that is Amatrol.
Right.
It was a windy day, and that is Amatrol. That's not paraquat. That is Amatrol, because as we got closer, it was the wheat was purple. So yeah, no, Amatrol. Yeah, I know that. I put that photo up for several reasons, and that's another reason, Pete.
Yeah. We better keep going. We're just about out of time, Pete. So you might need to get these last couple of done.
Sure. Okay. So yeah, we're almost there. So this is some data from another fence line trial that we did, where we had in the untreated 4,000 seed heads per square metre. That's seed heads, and each of those have got about anywhere from 20 to 50 seeds. And so what we've gone there is going through with you spray your Uragan. So Uragan has caused a huge reduction in the number of seed heads. So we've got 98%. So using some of these alternative mode of action herbicides, you can get really good control of ryegrass from the fence lines. Some other pictures. That's glyphosate resistant, crop margins, again crop margins, headlands there. And a few slides here on testing. Just that you can test plants, you can test these guys like early tillering. You can test. I've tested plants with 50 tillers, because what we do is we split those tillers right apart and plant 1 or 2 tillers in a pot, throw the rest away, feed it to the rabble or the chooks, and allow that to regrow and spray that with herbicide. So I received samples with only 5 plants. The grower was interested to know that if those 5 plants were glyphosate resistant or not, then we can test that. That's no problem there as well. You can also test for seeds, end of the season, and that's just a slide of a table that you get in the report, which just shows you what herbicides you've selected, you've chosen, the rates, and also the survival and the rating to indicate how healthy the plants are. These are very healthy survivors, and these are weak survivors to clethodim. And that's it. I'll leave it there.
Righto, good on you, Pete. That's great info. Now, we are at the end of our allotted time, but I will keep going and answer a few questions, but of course we'll understand if people have to leave now. You're welcome to leave, but we do have a few questions here that I think would be worth going over. One question is, what is the optimum time to come back and double knock fire breaks with paraquat after glyphosate? So, and should we add residual partners like Uragan to glyphosate? I think you've I think you've just answered that. But this double knock timing, Pete, I mean, I've done double knock trials, and I was doing them over susceptible ryegrass because we didn't have any glyphosate resistant ryegrass then, and there is a slight difference between that double knock timing whether you've got susceptible or resistant ryegrass, isn't there?
Yeah, look, double knock, it can mean different things. If you're if you're talking about a fence line later in the season, you've got a lot of plant, assuming you've got a lot of ryegrass there, or where we control, if we do the paraquat double, did the paraquat double knock, which was 100% effective, where we came in with paraquat first time and we killed 90%, waited 2 weeks. That 2 weeks allowed any plants that had received less paraquat to reshoot, and then that second application just came through and just killed everything else because you'd removed 90% of the foliage, and so you could really get in there and kill. Glyphosate paraquat, you sort of want, now if you want to, what you can do there is you can apply your glyphosate, and this is in a fence line situation, try and get rid of as much of foliage as possible. There could be other species as well. If you can open up that canopy, 2 weeks later come in with paraquat and get good coverage and kill everything else. In the knockdown situation where we're really trying to target or prevent glyphosate resistance, coming 3 days, up to 3 days after, 3 to 5 days after with paraquat is the best timing, and the reason is if you come in to, like, glyphosate stresses ryegrass. If the ryegrass is glyphosate resistant, and when you first apply the glyphosate, the plants will stress. And what we talked about earlier with stressed weed is that you get less herbicide. Herbicides don't work as well. So if you wait after that sort of 5 to 7 days, the plant's starting to stress and they don't take up that second herbicide as effectively. Whereas if you come in within that sort of 3 to 4 day period, before the glyphosate has had a chance to stress the ryegrass, that ryegrass that would survive the glyphosate, the paraquat will get in and do the job. So there's different, depending what you're aiming at, the timing can differ.
But you reckon ideally 3 to 5 days?
Yep, absolutely.
Yeah. Okay, here's a question I like. It's a good question. In a Roundup ready crop, would you prefer 2 applications of 1 litre per hectare 4 weeks apart, or a single application of 2 litres per hectare?
Would the 2 litres be at the beginning of the first timing?
I guess not. I would guess the 2 litres would be close to that 6 leaf stage of the canola.
Yeah, that's good. It's difficult to answer because you know, environmental conditions can really play a part as well. But usually I'd probably go in, if you're still getting good coverage at that late timing, then the 2 litres would probably be better. But you know, there's a lot of factors. By that time, it started to stress out your canola a little bit as well. You've got some, but I'd probably go the higher rate, assuming you had optimum conditions at both of those timings. And the higher rate would probably be better, assuming you can get, you know, good coverage.
Righto, good one, Pete. Now, what about, there's a question here about spraying glyphosate at night in the cool versus spraying during the day. What do you think about glyphosate at night?
It's a herbicide that really translocates, and plants aren't doing very much at night time. So spraying, you know, early in the morning, or you know, during the daytime where the plant's actively, you know, the whole mechanics of the plant's working, is you will get better results than you will at night time.
Just
so Yep. We're just trying to optimize, you know, get everything right. And if a small difference can make a big difference in weed control, then, you know, trying to apply it at the best possible timing is ideal. And so, yeah, during the day where plants are happy, they're growing, that's where glyphosate is taken up a lot more readily.
Yeah, and Stoles has just put up a comment talking about surface temperature inversions at night. Not only is that bad for getting weed control, but it's bad because it can drift away and do damage somewhere else. So,
absolutely.
Yes. Yep. There is another comment about alliance, so paraquat and amitrol being good on fence lines, and I just think to follow up on the fence line, I don't know if you've tried that mix, but the other thing that I've heard people talk about is using that paraquat Urigan mix early in the season, around the break of the season or not long after the break, rather than waiting till we've got that big jungle in spring.
Oh, well, that's what I've been advocating. That is the best timing for it because, you know, if there's anything green, you take it out with your knockdown, and then your residual is going to work really well, keeping all those other weeds from germinating during that season, or at least, you know, leave very few species come through, very few individuals, which can then be tackled easily come end of the season.
Alrighty. There was one other question as well earlier on from Glenn Reithmuller saying that on the label for Ultramax, it talks about using a coarse to very coarse spray quality, and his question was coarse droplets he said too small, but I think he meant a coarse droplets too big to hit small ryegrass.
They're usually not. They're okay. Where we've done those, I've put up on Twitter some different spray patterns where we've done similar stuff with more coarser droplets. In other years, you still get, surprisingly, still get quite a good coverage. And where you're using those air inclusion nozzles, you get the explosive effect of the droplet because it's loaded with air. And so even though the droplets might be big, they're exploding and they're spreading that herbicide around. So it's still pretty good. It's very. You think that you're going to miss ryegrass, young ryegrass, but at the end of the day, if your pressure's right, it's going to do the job. You're not going to get too many, if any, ryegrass escaping. They would normally be hit by a finer type droplet.
Yeah, and I think the other part of the answer to that question is once you're at that 70, 75 litres per water or more, then you get enough coverage with coarse droplets. But yeah, we need coverage with whatever herbicide we're using, and water rate generally fixes most of the problems in my experience. Someone else might like to dispute that. But anyway, we might finish up there. Thank you very much, everyone. That's been a great webinar. Very good, interesting information from you, Pete, but also great participation from everyone. Thank you for all of your questions and your comments that came up. I've thrown to you for some answers, and you've helped out with answers as well. But thanks, everyone. And WeedSmart will be delivering a few more webinars later on in the year. Probably the next ones will be in August, September sometime, so keep an eye out for them, and we'll see you next time. Thanks very much, and thank you, Peter.
Yep, pleasure. Good season, everyone.